So, good morning, students. In the last class, we had discussed about the introduction parts of microbes in human welfare. Okay, today we will discuss what are the topics we had. I had told you that we have to discuss in this chapter. Means the first topic among that topic is microbes in microbes in household products. So let's see, microbes in household products. Okay, so here, as I told you, that microorganisms, some are harmful and some are beneficial to the human beings. How it is beneficial means the first thing we will study in the microbes in household products. See here, some of the microorganisms we will consume it as it is live only. We will consume it. So, but also that microorganisms are not going to harm to our body. That particular specifically that microorganisms are not going to cause any diseases or infections in the body, because they are causing, but they are beneficial to our body. Okay, so let's see how these microorganisms are used in the household products. So first, in these household products, which are that microbes are used and which are that household products we will see. Among that one, the first is the dairy products. What it is? The first is the dairy products. What are the dairy products in which the microorganisms are used, such as curd, yogurt, buttermilk, cheese, etc. So these are some names of the dairy products in which the microbes are used. First we will see about the curd. Okay, so this curd is prepared by certain microbes. As commonly you know how the curd is going to be prepared. Okay, see, first we will boil the milk, then we will keep that milk for to come down. The temperature should come down, that is around, it should not too hot, not too cold. Okay, then what we will do, we will add a 2 spoon of curd into that milk. After 6, 7 to 8 hours, you can see that that particular milk has converted into the curd. What is the process is going to take place in the preparation of curd in that particular milk? Why that milk is changed into the curd? So what is that means? Here, see, majority of dairy products are prepared by, or majority of dairy products are prepared by one common bacteria is there. The name of that bacteria is LAB. What is that LAB means? That LAB means lactic acid bacteria. What it is? That it is the lactic acid bacteria. So these bacteria are used in majority of dairy products. Used in majority of dairy products. So let's see what are the particular dairy products in which, see, first of all, by name itself, this is the lactic acid bacteria. Lactic acid. Okay, this bacteria produces certain acids. Okay, what are that acids means? It produces the organic acids. What does it produces? It produces the organic acids. So let's see how these lactic acid bacteria are useful or beneficial for the preparation of the dairy product. Dairy product, which is one of the household product. So let's see here. see about the curd. Okay, curd, it is prepared by the inoculation of, it is prepared by the inoculation of a bacteria. The name of that bacteria is Lactobacillus. The name of that bacteria is Lactobacillus. Bacillus means which is one of the rod-shaped bacteria. Okay, Lactobacillus acidophilus. Okay, Lactobacillus acidophilus. What is that bacteria which is used in the preparation of curd mixture? That is a strain of lactic acid bacteria. This is also a strain of lactic acid bacteria, because here it is a lacto. Lacto means it belongs to the which category? That is to the lactic acid bacteria. Lactobacillus acidophilus. The scientific name of that particular bacteria which is used in the preparation of curd, that is the Lactobacillus bacteria. Here the LAB, that is the whatever the bacteria belongs to the lactic acid bacteria, that LAB what it will do in this particular, see, as I told you that particular how the curd is prepared is first what you will take the container, then you will put the milk, then you will heat at 100 degrees Celsius or above that also you will heat. Then you will take that container and then you will keep it to cool. Okay, it should cool down, the temperature should come down. Then what you will do means after cooling you will add in inoculum. Inoculum here means a spoon of inoculum. Inoculum here means a spoon of curd. What we are going to add? That is a spoon of curd. This curd, whatever you are adding a spoon of curd to that milk, this is the milk. Here temperature is 100. After cooling that, the temperature will come to the up to 40 to 50 degrees Celsius. At that time, what you will do means, you will just add the inoculum, a spoon of curd to that particular milk. At that time, this curd, a spoon of curd, will have a million number of bacteria. It will, in that spoon of curd, have the million number of bacteria. And which are the bacteria? The bacteria are the Lactobacillus acidophilus. So here, what happens means after adding a spoon of curd into this milk, whatever the bacteria which is present in this curd, that bacteria are the lactic acid bacteria. This lactic acid bacteria, the first thing what it will do after adding, what is the change is going to occur after adding into the milk means it produces a certain acids. It produces acids. Okay, what does this lactic acid bacteria will do? It produces the acids. Okay, that acid will coagulate whatever the acid which is produced by the lactic acid bacteria. This acid will coagulate. Coagulate means whatever the liquid nature of the milk it will be there, that is going to be coagulated. That means here you can see the milk is in a liquid form, totally in a liquid form. Next here what happens means whatever the milk is going to be there, it starts to coagulate like this. Means some solidness will come in the milk. That is known as the coagulation. It will start to coagulate, and whatever the proteins will be there in the milk. The second thing here, and partially digested the and partially digest the milk protein. Whatever the proteins will be there in the milk, that is going to be digested by the lactic acid bacteria. See here, the two important things of the lactic acid bacteria when it is added to the milk, that it is, first thing, it produces the acids. This is the first point. Okay, that acids will coagulate and partially digest the milk. This is the second thing. After producing the acid, first it will coagulate the milk. Then second thing, it will partially digest the milk proteins. Okay, in this way. And what is the temperature which is maintained over here in the preparation of curd? Whatever this coagulation, digestion of the milk protein, and production of the acids are there, that all things are going to be done. The temperature should be at 40 to 50 degrees Celsius. Okay, what that is 40 to 50 degrees Celsius of what? Of milk. The temperature should be maintained at what? 40 to 50 degrees. If this temperature is not there of the milk, if it is higher than this, if it is lower than this, the lactic acid bacteria will be not in an active form. They will not produce any acids. Okay, if there is no production in the acids means there will be coagulation, but there will be no formation of the or whatever the digestion of the milk protein will be there, that is not going to take place. So it will not that is not the correct preparation of what? Curd. So this is about the curd preparation. See here, first heating the milk at 100 degrees Celsius, then let it cool. When the milk should come at the 40 to 50 degrees Celsius, then you can add an inoculum. Here the inoculum means that is the spoonful of curd. That curd will have already a million number of what? Bacteria. What are the bacteria? Means that is the Lactobacillus acidophilus. It will belong to the group of bacteria that is known as the lactic acid bacteria. So what this bacteria will do in the after adding a spoonful of curd into the milk, what happens means first it produces the bacteria will produce the acid. Okay, then that acids will coagulate. Okay, that acids will coagulate the milk. Then in them whatever the milk proteins will be there, that is going to be partially digested, and whatever the milk temperature should be there, that it should be 40 to 50 degrees Celsius. Okay, so this is about the preparation of what? Curd. So this lactic acid bacteria, how it is beneficial? When we are eating the curd, at that time, the lactic acid bacteria, we are going to consume it live. They are not died. Okay, they are live, but also we are going to consume it. So what happens? How it is beneficial to our body? Let's see. Here, the lactic acid bacteria, which is present in the milk or in the curd, first of all, what it will do means it improves the nutritional quality of the improves the nutritional quality of the milk. How it is going to improve the nutritional quality of the milk means by increasing the amount of, or by increasing it improves the nutritional quality of the milk. How means as it contains certain organic acids and it also contains the vitamin B12. As it contains organic acids, that is one of the organic acid, that is the lactic acid. Here, organic acids and vitamin B12. Okay, these are the things which are present in the organic acids and vitamin B12. In this way, it is going to improve the nutritional quality of the milk. Who is going to improve the nutritional quality of milk? During the preparation of curd, this lactic acid bacteria, when you are going to add a spoonful of curd to the milk, it produces the acids as well as it also improves the nutritional quality of milk. How means it contains certain organic acid and vitamin B12, which is required to our body for better growth and development. Except that one, in curd or whatever the lactic acid bacteria will be there, it prevents. See here, lactic acid bacteria prevents the growth of disease causing organism Disease causing organism here, it means that it is the pathogens. What it is? That it is the pathogens. So, in our, where it is going to prevent the growth, it will stop the growth of what? Certain organisms which are causing the diseases. This bacteria, lactic acid bacteria, will enter into the stomach and there it is going to defeat them. Okay, they are going to kill, they are going to prevent. How they are going to kill them or how they are going to throw away from the body means by inhibiting their growth. They will stop their growth. Okay, of what? Of specially of disease causing organism. What are the disease causing organisms? That is the pathogens. Okay, this is one thing about the lactic acid bacteria, as well as it will also improve the microbial floral health of intestine. Okay, so here whatever the lactic acid bacteria is there, first thing it improves the nutritional quality of milk by producing what? By producing the certain organic acids and vitamin B12. Next thing, it prevents the growth of disease causing organism, that is the pathogens. Where? In the stomach. In stomach, it prevents the growth of pathogens. The last thing, it also helpful, or it is also going to cause the benefits for whom? For the next part of the digestive system, that is the intestine. See, whatever the constipation disorders of the digestion is there, that is constipation, diarrhea, or whatever the acidity. For that reason, many people, or whatever the doctors will suggest to eat you means, that is the curd. Why they are suggesting? For that reason, that to eat the curd means, as it contains the lactic acid bacteria, they will keep the health of the intestine as well as the stomach in a good manner. Okay, or it also improves the health of the intestine and the stomach. Whatever the diseases or disorders related with the digestive system is there, that all that problem can be solved by eating the curd. Okay, if there is an indigestion, gastric production, constipation, diarrhea, bowel movement, for all that reason, if curd is eaten after eating the meal, or simply you can eat the curd, then all that problems can be solved. Okay, in that way, they are telling it improves the microbial flora of what? Of the intestine. So this is about the curd preparation and their importance. Okay, next we will go for the next dairy product which is discussed in the NCERT book, that is the cheese. Okay. Next, that is cheese. I think you had seen the cheese. Okay, cheese which is used in the preparation of pizza and all. Okay, so that cheese, how it is prepared? Cheese, it is also one of the dairy product. What is cheese means? The curd is separated from the liquid part. Curd is separated from the liquid part, and that liquid part we can call it as the whey. What is whey? So to form cheese. See, when the curd is prepared, at that time, this is the container in which the curd is present. This is the curd. Okay. Then here you can see whatever the curdlings or the solid part will be there of the curd, it is settled down, and some liquid part will be there after preparation of curd. This is the liquid part of the curd. This is the curd, and this is the whey. Solid part we are calling it as the curd, and the liquid part here, liquid part we are calling it as the, what it is, that is the whey. So what they are telling, how the cheese is formed or how the cheese is prepared. Cheese, which is one of the household product as well as the dairy product, separated from the liquid part. This curd is separated totally from the liquid part, that liquid or whatever the whey is there, that is removed from the curd. Okay, and whatever the solid part is there, that is the pure curd will be there. By taking that one, they are going to prepare the cheese. So let's see about the cheese. Here, the cheese, depending on the water content, the curd will have a water content. Depending upon the water content, see here, depending upon the water content, the cheese will be soft, semi-hard or hard. Soft, semi-hard, and one more type, that is the hard. Okay, soft means what? In what we are calling it as the soft means. So here I told you, depending upon the water content, whatever the cheese will, whatever the curd they are taking, which is separated totally from the whey. So the curd will have a somewhat some water quantity or some water content. So depending upon that, this cheese is divided, or this cheese are of three types, that is soft, semi-hard and hard. So if it is in that particular cheese, the water content is around 50 to 80%. 50 to 80%. Then the cheese will be soft, very, very soft. Then if the water content will be 45% in the particular cheese, then it is known as the semi-hard. Not too soft, not too hard. Okay, that is the semi-hard. And if it is less than 40%, less than 40%, then we can say it as the harder cheese or the hard cheese. So one more thing, that is raw cheese is prepared by curdling of milk by lactic acid bacteria. Here the cheese are of two types, that is the ripened cheese and the unripened cheese. So what I mean by this ripened cheese and unripened cheese? First we will see about the unripened cheese. See here, the cheese are of two types, that is the unripened and ripened. Two types are there. What are that means? Unripened cheese and one more is ripened cheese. So first we will see about the unripened cheese. Here you know the meaning of ripened, how the fruit is going to ripen. You can take an example. See, when a mango is taken from the tree, okay, you will see the color of the mango. If the color is totally yellow in color, you will say that mango is ripened one. If it is green, then you will say it is unripened. So here also the cheese are two types, that is unripened and ripened. So here, whatever the unripened cheese is there, which is also known as the cottage cheese. What it is known as? It is also known as the cottage cheese. Okay, here this cottage cheese is prepared by inoculating. It is prepared by inoculating skimmed milk with, prepared by inoculating skimmed milk with culture. What is that cheese culture? Means a particular cheese which has a certain strains of bacteria. What are that bacteria? Means that here in the preparation of cheese, not only the bacteria are used, some fungi are also used in the preparation of cheese. What are that? Means that it is lactobacillus, lactobacillus, acetobacter, acetobacter, and next that it is rhizopus, then one more that is Saccharomyces. Okay, see here the unripened cheese or the cottage cheese. First of all, it is prepared by inoculating. Here they are going to add some culture into the cheese. Okay, to which cheese? That is the unripened cheese. That culture will have certain strains of microbes or the microorganisms which are used in the preparation of cheese, that is lactobacillus acetobacter. These two are the bacteria and rhizopus and Saccharomyces. These two are the fungi. Okay, here the bacteria which is used in the preparation of cheese, lactobacillus and acetobacter, and fungi is also used in the preparation of cheese, rhizopus and Saccharomyces. They are going to add a certain strains, certain bacteria or the fungi into the cheese in the preparation of in that particular curd. Here that is skimmed milk, what we are calling. In that they are going to add a culture of this anyone, not all, anyone, either the lactobacillus, either the acetobacter, either the rhizopus or Saccharomyces. So after that, it is going to, and one more thing, they are also going to add one enzyme that is very important, and the name of that enzyme is renin. What is that enzyme which is used in the preparation of cheese? That it is the renin. Then next one, this is about the unripened cheese. Next you will see how this unripened cheese is going to ripen. That is the second type of the cheese. That is the ripened cheese. You will see now. See here, ripened cheese. Here, the ripened cheese, it is prepared from unripened cheese. It is prepared from unripened cheese. How means so? That is by inoculating the different strains of bacteria and fungi. Okay, what they are inoculating into the unripened cheese means that is here certain process is going to do. They are going to dip, wipe, and then they are going to mature. Okay, this is the process in when the unripened cheese is going to taken for the what, for the preparation of ripened cheese. What are that means? That is dipping, then wiping, and then maturing. See here, When the unripened cheese is taken for the preparation of ripened cheese, here certain process they are going to implement. What are that means? Dipping, wiping, and maturing. Maturing by what? By the different strains of bacteria and certain fungi. And what are the different strains of fungi and bacteria? Means, see, whatever the cheese which has to taken for the whatever the cheese that is the unripened cheese is taken for the preparation of ripened cheese, it will take around 1 to 16 months. To convert it from unripened to ripened one, it will take how much time? That it will take 1 to 16 months. Okay. Then what are that particular? And this ripened cheese, one of the example of the ripened cheese is there. The name of that ripened cheese is the example of that ripened cheese is the Swiss cheese. So let's see about this Swiss cheese. An example is there. Swiss cheese. Here, in this Swiss cheese, you will find a large holes. Okay, large holes. Why these large holes are obtained in the Swiss cheese? Swiss cheese, one of the ripened cheese. Why there is a large holes in the Swiss cheese? That is due to by the carbon dioxide producing. There is a presence of certain carbon dioxide producing bacteria. What it is? There is a production of what? Carbon dioxide. See, why there is a large holes means due to the production of carbon dioxide. For what? A carbon dioxide is going to produce into that cheese. That's why you can see, for example, I will tell you, here this is the piece of cheese. You can see certain holes in the cheese. Why these holes are formed or obtained here means that is due to the production of carbon dioxide. So what is that particular bacteria which is responsible for the production of carbon dioxide means the name of that bacteria is Propionibacterium. Propionibacterium. Okay, Propionibacterium shermani. What it is? Propionibacterium shermani. This is the bacteria, name of the bacteria, which is used in the which is meant for the carbon dioxide production. Propionibacterium shermani. Then here some other fungi are also used, and some other examples are there of this ripened cheese. That is one more example, Rocky 40 cheese. What it is? That is the Rocky 40 cheese. And what is that particular, which bacterial fungi which is used over here means in the preparation of Rocky 40 cheese. These are the names of cheese. These are not the names of any bacterial fungi. These are the names of the cheese. In this preparation of Rocky 40 cheese, a fungi is used, that is the Penicillium Rocky 40. Penicillium Rocky 40. Okay, this is prepared by the bacteria, which is that cheese, Swiss cheese, which forms the large holes in the cheese. that is the Propionobacterium shermani, and here Roquefort cheese prepared by the fungi. And what it is? That is the Penicillium roqueforti. Okay, this is about the cheese. Some other household products are there, such as bread, idli, upma, dosa, or one more drink is there, that is the toddy. That I will discuss in the next class. Okay, thank you. If any doubt is there, you can ask.